Good afternoon. Thank you for attending. I'm Tom O'Hare, Associate Director of Sales for East Coast at Covaris. And today we'll give you a look into all that we could do with our sample prep offerings at Covaris. Historically, Covaris has been known for being the gold standard in DNA shearing. And we still are, however, we're much more than just shearing. We have the ability to provide solutions for all sample prep needs across different omics. And you could think of Covaris as the best choice for preparing your samples for any testing application. Covaris sample prep offerings are made possible by our patented adaptive focus acoustics, or AFA for short. AFA is a tunable acoustic energy that allows the ability to create energy that is adjustable across different applications. And that allows us the versatility to process samples across different sample types and different applications for various omics requirements. So you can really think of us as your solution for anything that you need in the sample prep world. Within workflows, AFA is utilized within multiple steps. So in this diagram here, you can see a few examples of some workflows. And the colored sections, so like extraction, for example, here, is one of the areas that AFA is used. So in an NGS workflow, AFA is used to provide high quality extraction and shearing going into a library prep workflow. And in protein, on the step down here, AFA is used to provide better extraction, purification, and digest going into mass spec workflows. And then in several other omics, we're able to enhance the extraction and enhance some of the other processing steps. Everything that we do at Covaris is about improving sections of the workflow. And some of the key advantages that we always focus on is improving performance, improving economics, and improving ease of use. So in genomics, when we're talking about shearing, what we're doing is we're providing you high quality, high yield, high throughput while saving cost on the back end. Our shearing is compatible with various library prep kits, which allows for versatility and allows for enabling automation. Non-AFA options generally have very long optimization and QC steps, high duplication rate, and high failure rates. However, our data shows a 98% pass rate, lower duplication rate, and no base pair bias, which leads to improved sequencing metrics. Our extraction, on the other hand, provides high quality DNA and RNA from low input FFPE, which allows for improving sequencing metrics and reduces the failure rate. So that in the end, improves your sequencing cost in the lab because you're not having to do reruns. Our FFP solutions also are automatable, which leads to reducing hands-on time, which can reduce human resources cost, and also reduces the financial burden in the lab. For protein, we have similar key advantages that impact the economics, ease of use, and performance. Some of those highlights are having greater total quantity yields of protein and peptides, up to an 18-hour faster protocol, so you can get your projects done in a single day. Our reproducibility is generally better than the alternatives that also are on the market. And we can provide faster digestion times without seeing increased miscleavages, which is a really nice advantage. And then we have some low sample input single plate workflows that we can offer. All of these things yield robust, reliable, reproducible protocols from any matrix. To quickly highlight some of the other advantages across some of the other omics, for epigenomics, we have more consistent shearing, we have better ability to reverse the cross-linking, and we can enhance your IP steps, making your IP either faster or more robust. And then for transcriptomics, we have better consistency of RNA shearing, and this consistency of RNA shearing actually yields improved sequencing coverage. We're also able to get improved sequencing from lower RNA inputs. All of this is made possible by our wide variety of instruments, kits, and consumables that we offer. Our instruments allow the ability to have low throughput from one sample to a handful of samples, up to 96 well and 34 well processing. So we really have an option for all the different labs out there. All of our instruments are uniquely designed to be either a benchtop all-in-one or fully integrated on a liquid handler to enable automation. Our kit offerings enable FFP, chip, and protein extraction and are designed to provide consistent, reliable results. Our team is more than happy to help you with anything from tissue processing to DNA shearing to protein extraction. Please just reach out anytime and we'd be happy to help you with your challenging protocols. So to end on reminding everyone, what I said is Covaris is the gold standard in DNA shearing, and we should really be thought of beyond that as your best partner for enabling improved results when results are really important to you. I would think of us as someone who's going to help you with that. And our solutions reduce hands-on time and financial burden to the lab. And that's really important for providing economics that are important to the lab. And our instruments can handle all your omics needs. So all of our instruments are built to work across the different omics. So if you had a lab that was doing genomics and proteomics, you could do both of those on the same instrument in the same lab. And we're here to help you with all that you need across your sample preparation challenges. All you have to do is reach out to any of us. We'll be more than happy to dive in as to how we can improve your results, how we can help you, and how we could talk about improving your protocols. Next, we have the pleasure of hearing Dr. Eugenio Divizio, our VP of Solutions at Covaris, present on germline and somatic solutions from Covaris. I thank you for your time, and we'll take questions at the end of all the presentations. Thank you.